This is a recorded video presentation on the topic immune response to infectious diseases. Both innate and adaptive immunity play an active role in the defense against microbes. Microbes may be able to evade or resist the protective defenses and be able to survive, thus causing infection. Injury of the tissues and disease may result from the survival when they overcome the host defense. Disease-causing microorganisms that we refer to as pathogens may vary from bacteria, viruses, parasites, and fungi. So the body is able to fight against these organisms through our innate immunity and adaptive immunity. However, sometimes they are able to evade and survive these um, defenses, host defenses. Our immune system responds differently depending on the site of entry, the route of spread, the tissue specificity, and the transmission, the manner of transmission. And again, we mentioned that the ability of the organism to evade and resist would be one of those important things to consider. The injury that may result from this invasion depends on the pathogenicity of the microbe and the immune system itself. The first of the microorganisms that can invade our body are the viruses. For the viruses, the main substance that is produced by cells are called interferon. So they are protein molecules produced by cells in response to viral infection. Most of the time, they block viral transcription and therefore they protect the body from infection. The presence of double-stranded DNA produced during viral replication will induce the production of interferons by the infected cells. Moreover, certain other cells are activated, for example, the monocytes, the macrophages, and the fibroblasts, to produce interferons. An example of an interferon is called interferon alpha, which inhibit the cellular replication of the viruses. Interferon alpha can activate the NK cells to directly kill virus-infected cells. Generally, interferons have no direct effect on the extracellular virus, and they can act in viral diseases before an antibody does. Its activity is not specific. NK cells, or natural killer cells, are able to destroy directly some virus-infected cells, and this does not need the presence of the MHC molecule. They can lyse virally infected cells. Humoral immune response is responsible for the production of antibodies. The antibodies against viruses provides protection against acute infection and reinfection. In viremia, antibodies are able to neutralize the virus. That means the presence of virus in the blood. It prevents the viruses to attach themselves to the receptor sites on susceptible cells. Examples of these viruses that are neutralized are the poliovirus, mumps virus, measles virus, and rubella virus. In cases of non-viremic infections like influenza, the presence of secretory IgA can neutralize the virus. Okay, the mechanisms by which these viruses are neutralized may be by aggregation of the virus and subsequent opsonization, and it can also be by the activation of the complement leading to lysis. Both mechanisms can act on virus-infected cells. Certain cells can act directly on the virus-infected cells like cytotoxic T cells, NK cells, activated macrophages. These CTLs or cytotoxic T cells can kill virus-infected cells by directly recognizing the viral antigen on the cell surface in association with MHC1. T helper cells can also be activated in the presence of the viral antigens and can release cytokines. The cytokines can attract and activate the macrophages to kill the virus-infected cells. NK cells can destroy the virus-infected cells during the early stages of infection, even before the appearance of antibodies. Antibody-dependent cell-mediated cytotoxicity, or ADCC, refers to when an antibody binds to the virus-infected cells and lead to lysis. Okay, this can be performed by NK cells, macrophages, and polymorphonuclears or neutrophils. How do viruses evade the host defenses? I'm going to mention several mechanisms and examples of viruses that are able to do these actions. Number one, they can overcome the antiviral effect of interferons. Okay, an example of this is hepatitis C virus. They can reduce the expression of the MHC1 molecule, and an example of this is adenoviruses and cytomegalovirus. Number three, they can also reduce MHC molecules, MHC2 molecules levels. Okay, examples would be measles virus, cytomegalovirus, and HIV. A large number of viruses can cause generalized immunosuppression. So that means that the immune system will be suppressed, leading to inability to respond to infection. Examples of these would be the mumps virus, measles virus, Epstein-Barr virus, CMV, cytomegalovirus, and human immunodeficiency virus. And number five is antigenic variation. So they are able to manifest themselves as a different um, substance. For example, influenza virus. In this table, we are able to see the different mechanisms by which our immune system carry out their functions against viruses. So it can be either humoral immune response or cell-mediated immune response. For humoral immune response, the secretions of different types of antibodies in coordination with a complement leads to the neutralization of viruses. On the other hand, for cell-mediated immune response, interferon gamma, which is secreted by T helper cells or cytotoxic T cells, can have a direct effect on the virus itself. It can also lead to the destruction of the viral infected cells, virus infected cells, and direct killing by certain NK cells and macrophages. Extracellular bacteria can trigger the activation of the complement. They can also activate phagocytes and they can induce the inflammatory response. On the other hand, they can also activate the humoral immune response to produce antibody, which is the main role. And then the second one would be the activation of cells that are responsible for their uh, neutralization, which is the lesser role. One of the substances that can be produced by bacteria is the toxins. These toxins can be neutralized through the antibodies produced by the body, the immune system. Another one, antibodies can also attach directly to the surface of the bacteria that can act as opsonins and can enhance the process of phagocytosis. Secondly, they can prevent the adherence of bacteria to their target cells because of the presence of IgA on mucosal surfaces. The activation of the complement can lead to lysis of the bacterial cell. These antibodies can also directly agglutinate bacteria, okay, and this can prevent the spread of these organisms and facilitate phagocytosis. As for cell-mediated immune response, microbes can be processed by antigen-presenting cells and presented to T helper cells. So this is possible by releasing cytokines that activate phagocytes and stimulate antibody production. They can also lead to induction of local inflammation. For intracellular bacteria, so bacteria that are found within the cells, innate immunity is not always effective. They can activate NK cells, and this can provide early defense against bacteria. But if you remember, NK cells mostly attack virus-infected cells. Intracellular bacterial infection tend to induce a self-mediated immune response, particularly delayed type hypersensitivity. So the presence of intracellular bacteria can lead to the production of cytokines produced by CD4 T cells like interferon gamma, and this can activate the macrophages to kill the pathogens more effectively. For the CD8 cells, which are also T cells, it can lead to direct lysis of the bacteria-infected cells. Sometimes there is a condition that may result from chronic T cell and macrophage stimulation. This can lead to the formation of granulomas. There are certain mechanisms that allow bacteria to escape from immune response. So this includes the presence of polysaccharide capsule that delays phagocytosis, the ability of bacteria to produce toxins, and their ability to cause disease, their ability to persist within the cells, and their ability to induce cell apoptosis. For example, Shigella flexneri. Furthermore, they can also block the action of lysosomes. For example, Mycobacterium. They can inactivate some components of the complement. They can induce the synthesis of actin fibers like Listeria and Shigella, enzymatic inhibition of active oxygen radicals like Staphylococcus aureus, and they can also hide from immune cells like in the epithelia. They have the ability to interfere with certain immune reactions. So that would be the mechanisms by which bacteria can escape the immune response. There are many important information that we can see in this table. So as you can see, there are three columns. So the first column is on the process of infection that is initiated by the invading microorganism, the bacteria. This is the host defense that is able to counter these infectious processes. And then thirdly, these are the mechanisms by which the bacteria are able to evade these mechanisms. Right, so the first one is attachment to host cells. So the presence of IgA type of antibodies can prevent these organisms from attaching themselves. However, some organisms are able to produce enzymes that can neutralize this IgA. Examples of these would be Neisseria meningitidis, Neisseria gonorrhoeae, and Haemophilus influenzae. So the pili of Neisseria gonorrhoeae is able to hide itself from being recognized by the immune system because they have antigenic variation. Proliferation, so this is the ability to multiply, is countered by phagocytosis or activation of the complement. So there are substances produced by organisms that resemble capsule, like the M protein, fibrin coat that inhibits phagocytosis or prevents phagocytosis. So there are also mechanisms that allow bacteria to survive within the phagocytic cells, and they can induce apoptosis in the macrophages. So the cells that ingest the microorganisms will simply die. Example is Shigella flexneri, I mentioned this earlier. For complement activation, so some gram-positive organisms can actually resist complement-mediated lysis, and they can insert membrane attack complex prevention in the long chain of the cell wall by lipopolysaccharide, which is present in some gram-negative bacteria. For invasion of host tissues, so there may be agglutination, direct agglutination by certain antibodies. However, there are certain enzymes that can inactivate certain complements that is present in some bacteria, for example, Pseudomonas. For toxin production, so there are certain antitoxins that can be produced by the body. However, the presence of again enzymes in some bacteria can prevents the bacteria from that promotes invasion of bacteria. So they can destroy the host tissue using this enzyme. As you can see, so bacteria are able to persist in causing infection by countering the host defenses, immune system. The next organism would be the fungi. Most of the time, fungi can cause superficial infection, for example, ringworm. However, some of them can also enter the system within our body through the lungs, for example, the spores of these fungi. The outcome would depend on the degree and type of immune response and may range from being unnoticed respiratory episode, for example, is like just simple coughing, to rapid fatal dissemination or even violent sensitivity reaction. Different forms of fungi would lead to different forms of defense mechanisms. In case of innate immunity, so two types of cells are generally activated, the neutrophils and the macrophages, so they perform phagocytosis, and this is very effective against candidiasis, candida albicans, and aspergillus flavus or aspergillosis. For acquired immunity, cell-mediated immune response is very similar to how our body reacts to intracellular bacteria. So this is also phagocytosis in general. Cell-mediated immunity predominates in the protection against Cryptococcus, Histoplasma, and Candida albicans infection. The general ways of survival would be similar to that of bacteria by providing capsules that prevent phagocytosis, resistance to digestion to macrophages, so mechanisms of surviving within the phagocytic cells, and destruction of the polymorphonuclears, maybe by apoptosis. Some yeast can activate complement via the alternative pathway. So the presence of the surface can activate the alternative pathway, but it doesn't have any known effect on survival. For protozoan infections, both humoral and cell-mediated immune response are also implicated. So humoral antibodies are found to be effective against the certain stages in the protozoal life cycle, except when they have already infected the host cells. So in this case, cell-mediated immunity is necessary. The mechanism is very similar to that of bacteria. The macrophages must first be activated by the T cells to enhance the killing mechanism, and the protozoa, like malaria, should trigger cell-mediated immune response. Large protozoa usually triggers antibody-mediated immune response. Some protozoans are capable of escaping the immune response. For example, Trypanosoma brucei has within them a glycoprotein coat that constantly changes. For example, another example would be Plasmodium can slough off their glycoprotein coat after the antibody has bound to it. So that prevents opsonization. For the helminths, the worms, they do not normally multiply inside the host cells, and because of the life cycle is complicated, only low-level immunity is induced. Helminths are generally attacked by antibody-mediated defenses. However, this may actually be ineffective. So CD4 T cells can play an important role in response to schistosoma infection, and cell-mediated immune response to helminthic parasites is mediated by Th2 cells that can stimulate IgE and activation of eosinophils that lead to the death of helminths. Generally are able to evade the immune response by disguising themselves to be host cells. So by evading the self from non-self. Okay, for example, in the case of schistosomes, they are able to absorb molecules from the host on their cell surfaces that prevents the body from recognizing them as foreign. Concomitant immunity, worms live in the host cells for many years and the body is unable to detect them as being foreign. So this sort of immune response is formed to prevent reinfection from the same worm. So that was the last slide. For this session, we talked about the different mechanisms by which the immune response is able to deal with microorganisms, different microorganisms, and also how microorganisms are able to evade and survive the immune response. Thank you.